Audio check. All right, guys, plugging away here. I have been having some intermittent, slow internet last few days, actually, at the house. I'm hoping that is not going to cause an issue today. I was just trying to watch NASCAR, and the how it looked on my screen, I'm definitely not pulling 4K quality there. So, all right. So we are getting into chapter 5 here. Now, I'm going to give you some important things to note about chapter 5. In my opinion, chapter 5 is the toughest chapter in the book. At least it is for me. It was for me when I took genetics. People that do well in the first 5 chapters and struggle with the later chapters in the book, in my opinion, are those individuals that had a really good experience in Bio 1, and they got really good background information and did very good in Bio 1. And because this is going to be new material, unless you've taken genetics before, most of this stuff is going to be completely new. And it's a little tricky. There's a lot of terminologies that you really need to nail down in order to work the more advanced problems. There are a number of advanced problems on the upcoming exam. Okay, so because chapter 5 the lecture got to you a little late, I did go ahead and extend the due date of the exam. I think I brought it out to, you'll have to check, but so it was originally, I think, the 11th. I think I extended it out to the 14th, whatever my announcement says, and whatever it says on Canvas, but pretty sure it was the 14th. However, that being said, my burden to teach you this stuff is obviously still there. But how I have everything scripted out in these PowerPoint slides does a job of its own in teaching it to you and walking you through the process. So it's just a kind of a preliminary warning. With this chapter, you're going to note that I am reading a lot from the slides, and then I'll stop reading the slides, and then I'll try to explain it in a different form. But the reason for that is because how I have it laid out in these slides, in my opinion, is the best way to present the information word for word. That being said, also, when I teach this in person, I do in class go over a number of the problems at the end of the chapter, and it's very interactive with the students how I'm doing that. But I'm doing it in the same way that the solutions manual does it, how it presents it, because it's, in my opinion, also the clearest way to see how to solve that problem. And just because of that, I haven't been doing that in these PowerPoints, in the lectures, because a lot of it's just going to be reading, the information to you. It's right there. And I haven't, you guys must be doing really, really well looking at the problems at the end of the chapter. I applaud you for that. I haven't heard any negative feedback about that. But so I just want you to keep that in mind. If it seems like I'm reading a lot from these slides, especially in this chapter, it's because I am. And it's because I think I have the chapter organized and presented in such a way that's really optimal on how to present it and how to present it to you. And, the author does an excellent job in this chapter of doing that as well, how he's broken down it into the most important points and essential points. All right, so before we get started, I just want to make sure looks like the transcript is looking pretty good. Okay. All right. So I've got 2 brief videos in this PowerPoint presentation, and I'm going to just try to play them so we can watch them together. And I do have, but I'm not 100% on how that audio is going to work. So you may need to go back and watch those brief videos on your own, because I don't know for sure if the audio is going to transfer over to the recording. Okay, so also I do have a couple problems in this chapter there's one sample problem that we're going to go over, and then I'll hopefully get part 2 I don't know, I might even go right into part 2 and take a pause right before we get to 3-point mapping or something. At the end of the chapter, so every semester when I do this in person, I do a 3-point mapping exercise in class. And, you know, the way the exercise works in class, it's divided up into 3 parts, and I have the students do a little work, and then we go over it, blah, blah, blah. So there's a lot more back and forth, and it's going to be very awkward doing this as an empty presentation with no interactive audience. But still, it's such an important component to this chapter, the 3-point mapping. I do want to work you through an example of it. But again, you know, how it's worked for you in the PowerPoints is really not, there's not going to be any surprises with me talking about it. I'm not really going to be introducing any additional or new information. So this is going to get broken up into 2 lectures just because I'm going to be losing my voice here as we get through the first half of the chapter, okay? Okay, so yeah, last chapter, chapter 4, we're looking at, you know, kind of exceptions to Mendelian genetics and also sex linkage, right, which was not addressed by Mendel. Right? Thomas Hunt Morgan started to address that. But now in chapter 5, though, we're also looking at an additional exception to Mendelian genetics, which works around genetic linkage. Okay, so in order to understand linkage, you have to completely understand independent assortment. And that's why I have a little video on there in case I've done poorly at presenting it to you. It's basically the opposite of independent assortment. Right. So just to recap, Mendel showed us the principle of segregation, right? That alleles separate during meiosis. Okay. And he also showed us the law of independent assortment, which means that alleles at one location on the chromosome assort independently from alleles at other locations, right? So if you have blue eyes, that doesn't mean you're more likely to have brown skin. Okay. 2 different genes, they're not genetically linked. That's for independent assortment. However, linkage says that there is a preference in terms of how those genes assort, right? So one will assort at a higher percentage rate, you know, with another gene because it's close together on the chromosome, right? So now when we talked about it's the reason we bring forth this definition, this term here, recombination, is because we're going to use it a lot. We're going to calculate recombination frequencies, okay? But it's just basically alleles, you know, sorting and then recombining into a new combination to give a unique phenotype that the parents didn't have, okay? But so we're going to get deeper there into independent assortment real quick, and I do want to show you this video. I've scanned hundreds of them, and but dozens, and this was the best that I found, just simply showing independent assortment. This is something you should understand completely right now, but in case you don't, I do want to show this video, which, okay. Okay, so this is an important feature, right? So, and he didn't mention the term homologs, right? But these are obviously homologous pairs that are lining up with each other. But when we're dealing here with 2 different genes, you can have different alignments, right? So how they align up, which is completely random during metaphase, is then going to dictate how they separate out during anaphase. Again, guys, I have a feeling you're not getting the audio on this because I don't see the transcripts transcribing. So go back and just watch the video from the PowerPoint slide directly, okay? All right, so now the spindle's coming to grab the chromosomes, separating out the homologous pairs. Remember, independent assortment is happening during meiosis one. Pretty much all the variation steps, not all, but the major ones, and we're going to get into crossing over again in this chapter, are happening during meiosis one. All right, so equal percentages, right? Equal chances, equal probabilities is a feature of independent assortment because everything is assorting randomly. Okay. All right, so also want to touch on the definition of recombination, right? So recombination means that there is a new phenotypic combination. There's a new phenotype. So look at the cross here. We've got P generation, homozygous dominant for A, homozygous dominant for B, and homozygous recessive in this gamete, in this parent. And the gametes that it produce, you can only have big A and big B. You can only have little a and little b. And during fertilization, now look what has happened. We have a heterozygote. Okay, this is just review. You guys already know this, that heterozygote is a recombinant. Okay, because it's new. That's all. This is an important term as we move forward in this chapter. Okay, so now here's where we get to look at an exception to Mendel, and that is what genetic linkage is. Because genes that are located close together on the same chromosome, we say they are part of the same linkage group and that they are linked. Right? And what it means is if they're linked, is that they're going to travel together during meiosis. They don't assort independently. Okay. All right, I've got a little video just to kind of drill this in because it's a very important concept as well. Again, if the audio doesn't display, it's hard for me to tell. I can hear it, but you may not be able to hear it. Just go back and watch it. All right, so A and B, are said to be linked, right? Because they have assorted together. They've gone, not independently, but dependently. B is depending on A and A is depending on B. Now, what the video is going to do, it's going to take it a step further, and it's going to show how crossing over can actually restore that independent assortment. And that's, again, a big part of this chapter is looking at how that occurs. Crossing over allows for the reassortment of linked genes. Basically, it just reverses that exception. And that, in this chapter, that's going to be the most important thing, is how you're able to bounce from, independent assortment to linkage back to independent assortment. What are the probabilities that are associated with that, all the phenotypic outcomes, doing test crosses to analyze each of those, right? So the key to being successful in answering challenging problems about that is understanding, how you can bounce back between those three different scenarios. Okay, let's look at it more closely. All right, so just a little history here. Walter Sutton, he was the guy that came up with the chromosomal theory of inheritance, basically discovered that genes are located on these DNA structures on chromosomes. And then the work of these guys, Bateson, Saunders, and Punnett, they did a bunch of experiments that showed results different than independent assortment of alleles. And they used the sweet pea plant because they could look at some different genes that the phenotypes were easily observed in the progeny, and those were in fact linked, right? So not the same pea plant that Mendel looked at. This is a different version, the sweet pea plant. So these guys found genetic linkage, right? So let's look at their example here. So what they look at is flower color and pollen shape. Okay, so you can have purple or red flowers. You can have long grain pollen or round pollen. All right, so they do, just like Mendel did, they did true breeding for both, right? So you got purple and long grain, which I think they say the dominant here. Yep, they're dominant because you see them in the F1. And then the red and the round are recessive, okay? So you fertilize the P generation and you get 100% purple and long grain, okay? That proves the theory of dominance. But now, when they self-fertilize the F1 generation, look at the numbers that they get. 284 purple long grain, 21 of these purple, round, red long grain, and about 55 of these red and round. And right off the bat, you should be able to identify that this does not, in fact, correlate to a 9:3 to 3:1 ratio. Why does it not correlate to that? Well, that's because the gene for flower color and the gene for pollen shape are on the same chromosome, and when they segregate during meiosis, they move together. They don't assort independently, so you can't get the correct genetic variation that you would get with Mendel's example when genes do assort independently. So this is a non-independent assortment of these two genes, okay? No 9:3 to 3:1 ratio. All right, so now what though can happen, like I stated before, is that with crossing over, right, the crossing over can result in recombination, right? And as we move forward with this, especially when we're doing 3-point mapping, instead of using like pictures of flowers and this and that, and instead of also just using the letters and their capitalization as nomenclature, we're also going to use these line notations, which can basically indicate where the genes relatively are located on a chromosome. Okay, so this is the notation system we're going to use, and a little bit more about it. So you have a homologous pair when, so the line basically indicates a chromosome, and you have a homolog is when one chromosome has 2 dominant or when the other chromosome has 2 recessive, right? So 2 dominant, 2 recessive, you're dealing with a homologous pair. You'll see 2 lines and also just 1 line. It's the same thing, but 1 line means that we're looking at 2 chromosomes. This is 1 chromosome. This is the other chromosome, okay? The genes have to be in the same order, though. So all the A's are going to be in this spot, then the B's, and so on, C's and D's. And that also is indicating locus, right? So these are in the same locus. They're said to be allelic because of that. So they're alleles of each other. These are in the same locus as well. Okay. All right. All right. Now this is important. So there are 3 scenarios when linked genes segregate together. Okay. So, actually, there's 3 scenarios under any sort of segregation. You can have what we've been talking about before. What we've talked about before, you can have no linkage, which means that the genes assort independently. We've now shown to you you can have complete linkage, such as the example with the flower color and the pollen shape. Okay. And in general, though, this is a very rare event, and it's why it was difficult, why Mendel really didn't see it with his phenotypes. Why is it a rare event? Well, the number of genes that are in a genome is so great. You've got a lot of genes in the genome. The chances that 2 of them are very close together, the 2 that you're looking at, is a relatively low chance of that. That's why this was, something that was discovered after Mendel. It's pretty rare to see. That doesn't mean that it's going to be less seen, though, in your problems moving forward. You're going to still work you're going to still work problems that involve complete linkage. Don't think you can rule that out because it's not as common. It's just not as commonly seen in genetics, but it's commonly seen in genetics problems. Okay. All right. And then the scenario that we're going to spend the most time on is where you have like a combination of these. And that's where it gets a little more tricky. Where you have some linkage and also some assortment going on. And really the only way to do that is to start now looking at, obviously more than 1 gene, but even looking at 3 genes, all right? So when we get into like 3-point mapping and things like that, okay? All right. The key to being able to work these problems is all about the test cross. In fact, moving forward, when you see crosses done, I'd say 75% of the time, what you're looking at from here on out is in fact a test cross, because a test cross is a way to figure out the genotypes of 2 possible dominant phenotypes, right? So remember, the recessive phenotype is easy because the genotype has to be homozygous recessive. But when you're trying to discern the genotype of a dominant phenotype, you have 2 possibilities, right? You can have homozygous dominant or you can have heterozygous. So when you're trying to discern that, you do a test cross and it can show you that. I'm going to show you here why it does that. All right, let's just go into that problem a little more. So let's look at this. You cross a purple flower of unknown genotype, right? So now it's unknown genotype. Yes, we don't know exactly what it is, but we know the 2 possibilities that it could be. Because it's purple, it could be big P, or it could be heterozygous. And we cross that with homozygous recessive, showing the recessive phenotype, which is little p. That is always the cross. You're always crossing one of the unknown dominant phenotypes with homozygous recessive, right? And that makes it simple, right? Test cross are always an unknown homozygous dominant with the homozygous recessive, right? So over here, this is the homozygous dominant, big P. This is the heterozygous, and what do we see? Well, when you have 100% of the progeny that are purple, you know that one of the parents was homozygous dominant. When you start to see the recessive phenotype, you know the parent is heterozygous because it doesn't come out in this dominant phenotype, big P. All right. If the test cross results in any recessive offspring, your parent is heterozygous. If you only see the dominant phenotype in the offspring, the unknown parent is homozygous dominant. And a test cross is always one of these 2 dominant genotypes versus the homozygous recessive genotype. Okay. Again, so far, we're 13 slides into this chapter, and almost everything has really been reviewed. Okay, but that's about to change. Just want to go back on all those, on these three things that we can determine, right? So, these are like basically the two steps that you're going to take when you are working problems where you have to discern between linkage and independent assortment. You're going to run a test cross to do two things. The first thing you're going to use it to do is determine the ratio of progenies in the F2. The second thing you're going to do is you're going to use those ratios to determine what your scenario is of genetic linkage, in that the genes are completely linked, in that there's no linkage, or that there's linkage with some assortment. And again, that up until this part, I hope that you're fully understanding what you can almost predict the direction that this chapter is going to go, and we're now going to look at multiple examples and multiple different test crosses to evaluate these three scenarios. All right, so I think I added this in to the PowerPoint slides, maybe not. But I want to work through a little. No, this isn't the work through problem. Where's that? Yeah, that's coming up later. Okay, but this is kind of a sample problem. And problems, genetics problems are tough, man, because sometimes you just have to sit there for a minute and absorb the nomenclature and the abbreviations. And okay, we're looking at molted leaves. We're looking at plant height. Which one's dominant? Which one's recessive? This example is pretty similar. It's pretty simple, and the phenotypes are very different, so it makes it a little easier to understand. But of course, genetics problems are great at giving phenotypes that can start to get tricky and sound similar and make it more confusing. All right, so we're looking at tomatoes, right? They've got leaves and they've got fruit, and they're plants. And some are tall and some are dwarf. Okay, tall is the dominant, dwarf is the recessive, and their leaves can either have a molted appearance or they can be normal, right? Wild type. And the normal is dominant, molted is recessive. Okay. So when the genes are completely linked, you get no recombinants. That's one way you're looking at a scenario of genetic linkage. There's no recombinants in the progeny. Right? Let's look at this example. This exemplifies complete linkage. So we've got a parent here that is normal with normal leaves and tall. Obviously, we know he's normal leaves and tall, so he could either be homozygous dominant, right, or he could be heterozygous. And then we have an individual that is molted and dwarf, and he has to be recessive. Now, we're doing a test cross, okay? And when you're doing a test cross, you have to do both scenarios, right? You have to do the one that's homozygous dominant and the one that's heterozygous. All right, so I think what we're going to do here is the heterozygous first, yes. So we do the test cross, and then we look at the progeny. So look how the gametes form. You've got your M and your D, that chromosome segregates. Your little m, your little d, that chromosome segregates. 50% are each of those. And the gametes for the homozygous recessive, 100% are going to be little m, little d. Fertilization, you combine the gametes, right? So basically 50% of them are going to be little m, little d, little m, little d. And 50% of them are going to be big M, big D, and little m, little d. And look at how the progeny are. Half of them are normal leaves and tall, and half of them are molted leaves and dwarf. I don't see any trees here, any tomato plants that are molted leaves and tall. This one and this one are identical to the parents, okay? And because of that, there's no new recombinants. And if there's no new recombinants, then our example is of complete linkage. Okay. All right. So, and what is the ratio of that? Look at this down here. So, because this is, yeah. So, remember when you do, so, and I haven't shown a slide. I'm about to show you a slide on that. But if you remember back when we did test crosses for independent assortment, we didn't get the 9-to-3 to 3-to-1 ratio. Instead, we got a 25% ratio among all four different new F2 genotypes. So those had recombinants in it, though. Okay? This scenario has no recombinants, right? No new phenotypes, and that's why you see the 1-to-1 or 50-50 type of frequencies. Okay, complete linkage, no recombinants, no new phenotypes. All right, independent assortment, right? And this, we've seen this before. We did this back, and I think that might actually be an error. I don't think it's, I think page 62 is chapter 2. I don't know, my memory's getting the better of me, but I'm sure that page is correct. But we have covered this before. When we do a test cross, right, where we take the, this is our P generation here, homozygous dominant versus a homozygous recessive. You get an F1 generation that's heterozygous. And now we're going to test cross this. When we test cross this with this, our progeny was always 25% of each of those different phenotypes. All right. And if you see, you do have recombinants in instances where you're doing independent assortment. There's no linkage going on. It's completely Mendelian, right? What you would expect if you did F1 to F1, what would be that ratio? 9-to-3 to 3-to-1. But we're doing a test cross. So you have to know the percentages. And this is a percentage that you should memorize, right? When you see 25, 25, 25, 25, you know that you're looking at a Mendelian example that's undergoing a test cross. And because it's a Mendelian example, you have independent assortment with no linkage. Okay, that is something to memorize. Also, seeing this 50-50 with test crosses, you know you're looking at complete linkage. All right, so how does it get wacky? Well, I think as you can see, using test crosses, it's very easy to distinguish between complete linkage and independent assortment. And also, I just want to revisit, because I didn't state this, but you guys, if you're trying to do Punnett squares here for all this stuff, you're going to drive yourself crazy, right? You have to be using that branch diagram method, which at this point, I think it should be pretty clear, right? These are all represented equally at 25%. And because it's a test cross, you've only got one possible type of gamete since they're homozygous recessive. But you've got 4 different possible gametes with independent assortment and a heterozygous. Okay. All right. So be sure you're not doing Punnets. I mean, if you have to do a Punnett, go ahead and do it. But you should be using these branch diagram with the different ratios is 1/4, 1/3, things like that. Okay. All right. Now, to make things trickier, if you have an example where genes are linked, however, they undergo crossing over, well, that can restore the independent assortment ratios because it swaps the genes back to almost, in a way of like thinking where they should be in a Mendelian sense. It puts them back to where they would be in a scenario of independent assortment. Right? Just to revisit what crossing over is, right? It's an event that occurs in late prophase 1, right? And it's an enormous contributor to genetic variation. And it occurs when the homologs pair up and a piece of one chromosome overlaps with another at the location of the chiasma. And when they fuse and when they pull apart, they have now a new recombinant chromosome. Okay, and then meiosis 2 is the same, but you can see these chromosomes now are essentially new chromosomes because they've swapped information with their homolog. Okay, so All right, so let's look at this. Crossing over, this is going to be linked genes, and we know they're linked because look, A and B are going to be linked because they're on the same chromosome. Okay. When linked genes are crossed over, when genes are linked, but there's no crossing over that occurs, which is what's going on here, you don't have any recombinants, right? However, when genes are linked and crossing over occurs, now you have recombinants. You basically now made it so those genes are exhibiting independent assortment-like properties, right? You're getting recombinant genotypes. Okay, that's where that comes from. All right, so at this point, I'm just going to earmark this in my head. We're at 19. I just want to kind of revisit this very important thing. So you should fully now understand these three terms, right? So segregation, independent assortment, recombination. Where am I going here? That's not what I want. So I'm looking for this right here. Now you should understand the importance of these three scenarios, right? Complete linkage, no linkage, linked with some assortment, which is where you have linkage with crossing over, and those test crosses, these guys right here, right? Complete linkage, you get the 1-to-1 with the test cross. And when you have no linkage, you're basically doing what we did before, Mendelian independent assortment, you get the 25% all across with the test cross, right? The test cross is the F1 heterozygote crossed with the homozygous recessive. All right, okay. And finally, before we get into a sample problem, and then, yeah, that's everything for the chapter. We're going to do a sample problem, and we're going to learn like one sort of continued variation, cis and trans, and we're going to have a summary table, but that's all of it. So I think where the point of most students are at right now is they fully understand everything I've talked about. It just hasn't sunk in yet. And in order for it to sink in, you got to practice it. Practice it means writing this stuff out, just without a book assisting, trying to recapitulate these crosses in your mind. All right, another thing that you'll need to know how to do is calculate something called recombination frequency. So this basically looks at when you have an example where you have recombinants, it basically tells you the percentage that those recombinants represent among themselves and all of the other progeny that could be in a cross, right? So look at this cross here. We've got 4 different phenotypes, 4 different genotypes, right? And 4 different phenotypes. And we've got 55 of these and 53 of those, and 8 of these and 7 of those. And these here are unique. They're different than the parents, right? So the parents are normal leaf tall. There's your normal leaf tall. And the other parent is molted leaf dwarf. But there are no parents that are normal leaf dwarf. There are no parents that are molted leaf tall. So these represent the recombinants. So what percent is these of this? And that's what recombination frequency is. And it's a very easy calculation. You just take the number of the recombinant progeny divided by the total, multiplied by 100, right? And you get a percentage. Now, that value is important because from here on out, we're mostly going to work with recombination frequencies and map units and how that corresponds to map units on a chromosome to look at linkage and do crosses and things like that. And then also, it's important to understand that at a certain point, I'm telling you this too prematurely, but when you reach a recombination frequency of 50%, at that point, you could basically assume that the genes are not linked. So because you really can't calculate, it's one of like the shortcomings of using recombination frequencies. It's only kind of accurate. It's only useful up to a certain point. Okay, but I'll get into that later. Again, just at this point, understand that it's representing the percentage of new combinations of traits, right? So in this cross, we don't have, we get new phenotypes from this right down here, and those are the recombinants, and you can calculate recombination frequency. Okay. All right, let's look at this example. Silk moths. I don't know how to pronounce that. Bobomyx mori, whatever. I don't even know what a silk moth is. It's a caterpillar. I don't know. It's a bug. Like, whatever. All right, so we got red eyes, RE, and we got white banded wings, WB. It looks like right off the bat, those are the recessive traits. They're encoded by two mutant alleles that are recessive, okay, to those that produce the wild type traits, which are RE plus and WB plus. Okay, so remember, when WB is white banded, it describes the wings, and RE describes the eye color. Okay. All right, here's the problem here. Moth homozygous for red eyes and white banded wings. And at this point, I'm usually using a pencil and starting to write stuff down. But also, maybe without doing that, look at the problem and start to think about what we learned in this chapter and see how what we know here applies to what we've learned. Okay, so a moth homozygous for red eyes and white banded wings is crossed with a moth homozygous for the wild type traits. Okay, so we haven't done a test cross here. Looks like just a typical, P and F1. The F1 have normal eyes and normal wings. Normal eyes and normal wings. So here, the wild type trait has come out in the F1. You can assume it's 100%. All right, so now we're taking the F1, which are heterozygotes. So F1, let's make sure they are heterozygotes. They are, because we did a moth homozygous for red eyes and white banded wings crossed with the moth homozygous for wild type. Right. So the F1 are heterozygotes. Okay. The F1 are crossed with moths that have red eyes and white banded wings in a test cross. Now we're doing the test cross. And we can assume that the genotype, right, for the red eyes and white banded wings, the only way to get that phenotype is to be homozygous recessive. So just like we've done before, we're doing a heterozygote versus a homozygous recessive. And now look at these values. Look at the values that we have. So if it was a Mendelian situation, what would the ratios be? Well, remember, independent assortment, when you do a test cross of heterozygous versus homozygous recessive, you get 25% of each progeny. And that's not what we have here. So we don't have an example of independent assortment. But let's read on. Let's look at what phenotypic proportions would be expected if the genes for red eyes and white banded wings were located on different chromosomes. Oh, okay. Well, I think I just gave you the answer to that question. So it's what phenotypic proportions would be expected. Be sure to understand what we're looking at here. We're looking at a test cross. So if you were to say 9 to 3 to 3 to 1, right? I'm just checking to make sure that's the right answer. If you had said 9 to 3 to 3 to 1, that would be wrong, right? That's the anticipated ratio for a typical Mendelian cross where you're self-fertilizing the F1s. Here we're looking at test crosses. And like I said at the beginning of this lecture, almost 3 out of 4 times moving forward with this stuff, we're mostly looking at test crosses because they can figure out which dominant phenotype we're dealing with, whether it's homozygous dominant or heterozygous. Okay. What phenotypic proportions would be expected if the genes for red eyes and white banded wings were located on different chromosomes? That means no linkage. I'm going to jump to the next slide here. It's going to have a lot of spoiler alert. And it's going to have the answer. All right. So if the genes are located on different chromosomes, then we can assume independent assortment. Right? Because we are test crossing a homozygous moth with one that is homozygous recessive. So we will get the proportions one always gets with these test crosses. Couldn't have said it better myself. Right? You would get 25%. It's all these same phenotypes here, but instead of getting this, we would get 25% among all of them. Right? Typical independent assortment test cross results. Okay, what was the second question here? What is the percent recombination between the genes for red eyes and white banded wings? So the percent recombination, see how this is termed, percent recombination between the genes for red eyes, so between two different allelic pairs, right? What is the percent recombination? Well, we can see that we have recombinants and non-recombinants. I'm going to jump ahead just so I can get the different things out here. So now, how do you know which is which? Well, there's two ways to know. In this problem, there's definitely two ways to know. One way is you can look at this problem here and you look at the progeny and you see which ones are new recombinations, right? So just to melt my brain a little bit, you know, red eyes and white banded wings were there, right? No, we've got to look at the test cross. We've got to look at the test cross. So the test cross is that, right? Because the homozygous recessive. And it's also the heterozygous, right? But so it's these two. So the original parents of what we're seeing here are these: the moth homozygous for red eyes and white banded wings, red eyes and white banded rings, right there, is crossed with the moth homozygous for the wild type traits, right there. Okay, so those are your non-recombinants, right? Because they were present in the parents. And I like this problem because it makes you focus on the test cross and not all this crap up here. But these guys, the red eyes, wild type wings, the wild type eyes, white banded wings, are new phenotypes. So they must be the recombinants. These are the non-recombinants. They were present by the parents. These are the non-recombinants. They're new. That's one way to tell by comparing the progeny to the parents. The other way, usually, almost always, is looking at the numbers. The ones that are most common are going to be the non-recombinants, and the ones that are least common are going to be the recombinants. Okay? Let's go back over here to where the answer is. What is this up here? So this is the P generation. Okay, right. So this is, I'm just summing up the problem here, right? So here's the homozygous recessive. Here's the homozygous dominant and the wild type. Here's your heterozygous F1, and then here's the test cross, right? So here's your heterozygote crossed with the homozygous recessive, and then here's the results of the progeny. So again, these ones in red here are unique. They're not either of these. The ones that are purple and green are just like these. Not these, we don't care about those, like these. Okay, so now you know which ones are recombinants and non-recombinants, so you can calculate recombination frequency. You take the recombinants, 19 plus 16, and you divide that by the sum of all of these, which is 879. So you're taking 19 plus 16, whatever that is in my head, 20, 35, right? Divided by 879 times 100, and you have 4%. So, you know, the frequency between these two genes, red eyes, wild type, is 4%. You can say that. Okay, that is 98% of everything in the first half of this chapter. I'm going to take a break here in a sec. But there's just one little variation we want to throw on top of it. And that is that when linked genes segregate together and crossing over produces recombination between them, the genes can exist in two different orientations. They could be coupling, which is also called cis, or they can be in repulsion, which is also called trans. Okay? When in the coupling configuration, one chromosome contains both the wild types, the other chromosome contains both the mutants. In the trans configuration, you have the wild type and the mutant allele on the same chromosome. Now, maybe this doesn't mean anything to you yet, and that's fine. It will start to mean, and you'll be able to pick out if things are cis or trans quite easily when you start looking at scenarios like this. And this is the nomenclature I want you to start getting kind of used to, because this Drosophila manner of kind of showing genes and doing crosses on chromosomes is going to become more common. So, all right. So I also, before I go through this problem, I want to kind of jump ahead a little bit. If you look at the numbers of progeny and how they relate to non-recombinants and recombinants, you're going to see they're the same, right? They're going to have, they're not going to have the same recombination frequency, but they're going to have the same value for recombination frequencies, whether or not the genes are oriented in the cis or the trans. Here's cis coupling or trans orientation. What changes is which combinations are considered non-recombinant and which combinations are considered recombinant. Okay, because if you compare this, like these all values are the same here, green, brown, now we're looking, look, things are different here, green, black, purple, brown, so things are flipped. Green, brown, green, black, purple, black, now we're doing purple, brown, okay? It's flipped, and that's really all it is. With cis and trans configurations, the combinations, the types of recombinants, it's the best way to word it. The types of recombinants are flipped, but the ratios are going to be the same. All right, we're doing test crosses here. This is a test cross for the cis. This is a test cross for the trans. So in a cis, the alleles, wild type alleles, are on the same chromosome. The mutant alleles are on the same chromosome. Trans, wild type and a mutant are on the same chromosome. Wild type and mutant are on the same chromosome. We're going to test cross these, which means that we're going to cross it with a homozygous recessive. All right, and in homozygous recessive, it doesn't, you can't be in cis or trans because it's the same thing on both chromosomes. Right? You only have homozygous, I mean, you only have cis and trans situations when you have heterozygotes. Okay. All right. So if you lay this out, make the gametes, right? Make the gametes, and then you do the combinations. This is how everything comes together right here. Right? And it comes together with the same recombination frequencies, but how those recombinants are set up are obviously going to be different because, even though the test cross has the same phenotypes, that's the other distinguishing difference between cis and trans. Here you've got an organism that's green thorax brown. This guy is green thorax brown. It's just because it's flipped on different chromosomes. But because these alleles are segregating, it can change the way that the groupings are situated. Okay, this is important. There's going to be a few questions that recognize this, and as you work in the problems out, you'll see this flipping that occurs in the numbers. All right. This table. You see, I've got everything highlighted on this table. Well, that should be a clue that it is an extremely important table. But this is not a table you memorize. This is you should know everything on this table, but you should understand the information in this chapter to be able to derive all of these frequencies. All right, and it's just a review of all the stuff we looked at. So here we've got independent assortment, and we're doing a test cross, the heterozygote versus the homozygous recessive. What are the different results, the recombinant, the percentages, the frequencies in scenarios that you have complete linkage, you have no linkage, and you have linkage with some crossing over? Well, we know that if you have independent assortment, you're going to have 25% of each of the different ones, and you're going to have recombinants and non-recombinants. In examples with complete linkage, you are going to have no recombinants, and it's going to be a 1-to-1. Remember I told you that those two figures, bam and bam, are very important. Got to know those. Oh, it's been an hour. Okay. And then when you have linkage with some crossing over, right, you're going to have a combination of recombinants and non-recombinants. And the non-recombinants are going to be the major players in the population. They're going to be the most numerous. They're going to have recombination frequencies greater than 50%. The recombinants are going to be less than 50%. Now, why does it just say everything is in coupling here? It's all in cis. Well, if it was all in trans, it would be the same numbers. Your non-recombinants would still be more than 50%. Your recombinants would still be less than 50% if this was in trans. The only thing that would be different is something this table cannot show, and that is how these are grouped, the non-recombinants, recombinants, and how they're grouped together. Okay? Again, this is not something you memorize. This is something, after fully understanding this, you don't need this table because you know it. You know these ratios, you know what these things mean, these three different groups, you know what recombinants are, you know how to calculate recombination frequency, you know the difference between cis and trans, you know that we're dealing with test crosses here, right? We're not looking at 9 to 3 to 3 to 1 stuff. This is test crosses, which means 25% across the board for Mendelian and 1-to-1 for linkage, and then you know various values for linkage with some crossing over, obviously, but the non-recombinants are always going to be more than 50%. The greater population, the greater number of individuals in the progeny, and these are going to be less, okay? All right, that's a book. I think I got one more slide here. Yes. So this is just going to take, I might actually keep going here a little. No, I should probably cut it off. All right. So now, this is not too bad. This is not going to make it more complicated. All right. So, you know, geneticists don't really use recombinant, like the value recombination frequency very much. They use different units like called a map unit or a centimorgan. But it's the same thing. One map unit is equivalent to 1% recombination rate. Okay? And the next part of this chapter, what we're going to do is we're going to be able to see these different frequencies and be able to map genes on a chromosome. For example, if you have 3 genes, you can determine which gene is in the middle based on the distances between all of those genes. Right? It's not rocket surgery here. And that, as we approach this in the next part of the chapter, and I'm going to jump right into it here. I'm going to log off, save the file, and jump right into it. But as we're getting into that second part of the chapter, that's your end. Well, not your end point, but that's where you want to go first. You want to find what is the middle gene. All right, so keep that in mind here for the rest of chapter 5. And I'm going to get a drink of water and save this file. And I will be back to finish chapter 5. Okay, see you guys soon here. Not recording.